Hello everyone. Welcome to another Heart Muscle Talk session. We have a great speaker today, and the session will be chaired by Walsh from UCL and Barts in London. So over to you, Luis.
Thank you, Alex. Hello, everyone. It's a pleasure for us to have today Dr. Roddy Walsh from Amsterdam. He's an expert in cardiogenomics and will talk to us about the complex genomics of cardiomyopathies. Thank you very much, Roddy.
Great. Thanks, Luis. Just share my screen.
Looks good.
Okay. Yeah. So thanks very much for inviting me to speak today. So I'll talk about some recent work I've been doing exploring the complex spectrum of Mendelian genetics and cardiomyopathies. And I hope to convince you that maybe kind of exploring some of the rarer genotypes and cardiomyopathies is quite important. And also the importance of perhaps expanding research into understudied populations. So as you probably know, we've been doing research into genetics of cardiomyopathies for probably about 35 years now. In fact, this year it's 35 years since the seminal paper was first published by the Seidmans, which found the first gene which was associated with cardiomyopathies. This was MYH7 associated with familial hypertrophic cardiomyopathy. Also, there's been a lot of research since then. I guess traditionally we would have viewed cardiomyopathies as Mendelian diseases of largely autosomal dominant inheritance. That might be what you would see in textbooks and reviews. But of course, we know these days that the genetics of these conditions is that much more complex than that. So in clinical genetic testing, we still only identify causal rare variants in about 20 to 40% of cases, which means that for most patients that we test, we don't detect a direct primary causal variant. We know that even when we do detect one of these variants, there's variable expressivity, of course, and incomplete penetrance. And now we know, of course, from biobank studies that are coming out now that there are many unaffected carriers of these pathogenic variants within the population. So as of most of genomics research has tended to be concentrated in individuals of European ancestry as well. And the case of cardiomyopathies probably has been concentrated in adult onset cases. So all of this could mean that recessive inheritance up until now has been fairly underreported. So I think all of this points to the fact that there are probably much more complex genetic and non-genetic etiologies underlying these diseases. So this graph just shows you an overview of the rate of discovery of monogenic causes of cardiomyopathies, an overview of when these genes were discovered, the timeline as to when these genes were first proposed. So here we're just showing what we consider now to be validated disease genes. So these are the ones given definitive, strong or moderate evidence by the ClinGen evaluation. So in red, we're showing genes with autosomal dominant inheritance linked to HCM, hypertrophic cardiomyopathy, and in blue, those with autosomal dominant inheritance linked to DCM or dilated cardiomyopathy. So I think one of the main things you can see from this is that in recent years, the rate of novel gene discovery has really slowed down quite a bit. Now, I think the rate of gene discovery has broadly gone through two phases for cardiomyopathies. So the first of these was linkage studies in the 90s and early 2000s. Where we took large family pedigrees and did segregation studies to find the genes and variants which were segregation with the phenotype. And largely these were very successful at identifying most of the main genes which are involved in these diseases. So this was followed by the candidate gene study era, and I think in general this is fairly less successful in identifying robustly associated genes. We've done many genes were implicated by these studies. I think probably the main reason for this is that our understanding of the genetic architecture of rare variation in the general population has improved a lot in recent years, particularly with the advent of databases like Nomad, where we found out that rare variation is much more common than we previously had considered. And recently, of course, we've had the ClinGen reevaluation of the gene to disease relationships for cardiomyopathies for HCM, DCM, and ARVC. These have found that about 2/3 of all the genes which have been implicated in the disease are now considered to have limited or disputed evidence. So I guess the question is, why is it so difficult for us to identify novel Mendelian disease genes these days for cardiomyopathies? So probably a number of reasons for this. So even though about 2/3 of our patients, we do not detect a pathogenic variant in clinical genetic testing, it's important to understand that for most of these, we now would consider that these are unlikely to be Mendelian causes of disease, but rather they have a much more complex etiology. So there's a number of reasons for this. I guess one of the main reasons being that these tend to be more sporadic cases without a known family history of disease. So we're beginning to explore this complex etiology a lot more now, particularly with genome-wide association studies. So some of the latest studies are shown here, which are available as preprints in medRxiv. And these are multi-center studies, which now include up to about 6,000 cases for HCM and DCM. So these have not only shown detected lots of genome-wide significance loci for cardiomyopathies, as shown in this figure here. They've also estimated that the overall heritability attributable to common variation is quite large, probably somewhere between 20 and 30%. And as expected, it's a bit larger in those cases where we don't detect a primary rare causal variant compared to those where we do. Nevertheless, I think there probably are still Mendelian genes left to discover. But of course, it's probably likely that these are fairly rarely causative of disease, probably even causal in less than 1% of cases each. And of course, this provides a lot of challenges with discovering these variants. So we're probably unlikely to detect, find such variants in large family pedigrees. One of the reasons for this, of course, is that most of these have already been resolved, certainly in Europe and North America. I think you'd have to be pretty lucky to find a large pedigree now where the causal variant is likely to be located in a novel gene. So the second way we can detect, obviously, novel Mendelian disease genes is through case control analysis, looking for enrichment of rare variants within the gene in your case cohorts versus controls. But of course, this is also quite difficult when you're looking at rare causes of disease. So as an example of this, and this is some data from a study which is currently in review. It's been led by Sean Jurgens here in Amsterdam with his colleagues at the Broad Institute. And they've combined data from 3 different biobanks, UK Biobank, all of us in the US, and the Massachusetts General Biobank, and a total of about 750,000 individuals there with genome exome sequencing. So with this, they've done rare variant association studies across 600 different disease phenotypes. And the data I'm showing here is this rare variant association study for primary or intrinsic cardiomyopathies as defined by an ICD codes. So across these 3 biobanks, they've got close to 14,000 cases, over 735,000 controls. As you can see from the data, so these are the significant hits, and those above genome-wide significance, which is that line here, which accounts for multiple testing correction across 20,000 genes. So I think what this data really shows nicely that we're now really powered to detect many of the known Mendelian disease genes for cardiomyopathies, as you would recognize these here, and shows you the power of these studies to really detect a lot of known disease genes. But what it also shows you, I think, is that we're still probably not powered enough to really use these type of approaches to detect novel Mendelian disease genes for cardiomyopathies at genome-wide significance. Now, it's likely that just below this genome-wide significance threshold, at more nominal levels of significance, we probably are detecting some novel disease genes. But of course, distinguishing these between potential false positive hits is quite tricky. And what we probably need there is some additional levels of evidence, and that's something I'll come back to a little later. So I think potentially the third challenge for novel Mendelian gene discovery in cardiomyopathies has been the lack of diversity in genomics research. So obviously our knowledge of a genetic architecture of a disease is going to reflect the populations we're analyzing. And I think in particular recessive genotypes are probably have been understudied until now, as these may be more prevalent in certain diverse patient groups. So these could be pediatric cases, and it could be patients within isolated populations where founder variants might be more prevalent, or also in regions of high consanguinity. Unfortunately, I think there's the recent years now, there's been a lot more research in this area. So we've had several studies now, cohort studies, looking at the genetic basis of childhood and pediatric onset cardiomyopathies. And also in recent years now, we're seeing a lot of gene-specific studies looking at biallelic variants, which may cause recessively inherited cardiomyopathies. So I think recessive inheritance like this is really potentially a really good source for novel genetic associations. So recessive pedigrees are really highly powered to detect novel associations even within fairly small family pedigrees. So this is because what we can do with these is unbiased genome-wide approaches with an affected child and 2 unaffected parents. So this is exome or genome sequencing, followed by various variant filtration methods to identify a small numbers of potentially plausible candidate variants. So these could be homozygous rare variants, coding variants in genes, which are found in the homozygous in the patient and heterozygous in the 2 parents, 2 unaffected parents. Or there could be compound heterozygous variants, that's 2 different rare coding variants within the same gene within the patient, where they inherit one of these from each patient, from each parent. So this various reasons why these are being increasingly applied now for diseases like cardiomyopathies. There's obviously the reduced costs associated with exome genome sequencing, which makes it more feasible to do these studies. We're also, of course, moving research outside of the traditional centers of genomics research into more isolated populations and more diverse regions of high consanguinity. As I mentioned, we're also seeing a lot more research into the rarer pediatric forms of disease. There's still some challenges associated with this, of course, and these studies often tend to be small, looking at kind of single pedigrees or single case reports. You can have issues with this various filtering approach. You might not always detect the actual causal variants or prioritize the correct variant with your filtration approach. And of course, if there is a lack of replication, this may provide insufficient evidence to prove the association. So this was the basis of the kind of study we've recently done to try and explore this. And we wanted to look at all the recently published data and to do a meta-analysis to try and identify the robustly associated recessive cardiomyopathy disease genes. And then within these to try and explore the spectrum of dominance and recessiveness. So this study, the joint first authors in this study are Alex Lipov and Sean Jurgens here in Amsterdam, and also Francesco Mazzarotto in Brescia in Italy. I'll also mention some results of another study we've been doing in collaboration with the Mahdi Yacoub Research Network in Egypt, which explores the genetic architecture of hypertrophic cardiomyopathy in the Egyptian population. This is a study led by Dr. Mona Aluba. So in terms of the meta-analysis of the published literature and trying to identify these valid recessive genes, so for this, we did a PubMed search with a search for cardiomyopathy, terms of the cardiomyopathy phenotype and various terms around recessive inheritance, and detected about 2,000 reports which came up in this search. So we triaged these to find out relevant papers, which are human clinical genetic data with biallelic variants where they're associated with cardiomyopathy phenotypes. So the next step then was to try and identify and distinguish between those patients with a fairly isolated cardiomyopathy phenotype, or where cardiomyopathy was a primary clinical feature, from the kind of more syndromic forms. So it's not always easy to kind of really find a very clear distinction between these, but we did our best to try and focus on the most isolated cardiomyopathy phenotypes. So in total, we found 71 reports, which were describing 33 different genes, and mined various information on this related to study design and the genetics to see what kind of the strength of the evidence underlying these, and also various clinical data around the phenotype and the outcomes of the patients and their demographic details. So to define robustly associated gene-disease associations, so we define these as genes where we found biallelic variants, where there was proven recessive inheritance from the parents. And these were identified in multiple independent family pedigrees, where these variants were identified with unbiased genome-wide methods, that is exome or genome sequencing. Also, for a number of genes, we also noted whether there was a significant enrichment of biallelic variants in cases versus controls in large case controlled studies. Beyond this, we also looked for additional supporting evidence. So some of these family pedigrees were quite large, so we noted where there were strong blood scores. We also noted some additional biallelic cases where recessive inheritance mightn't have been proven, that is, the parents were untested, but it's still probably likely. Also made sure that for the implicated variants, that none of these were present in the homozygous state within NOMAD. And also looked for additional supporting evidence, for instance, whether these genes were putative causal genes at GWAS loci for relevant traits. For those genes where biallelic truncating variants were found within the human, within the patients, that is full human knockouts, also noted whether there were concordant phenotypes in mouse knockout data, and then also noted that for these genes, that there are no biallelic truncating variants at all in these genes in databases like NOMAD or UK Biobank. So in total, we found 18 genes associated with isolated cardiomyopathy with recessive inheritance that we consider to be robust or replicated. So we plotted this onto the same timeline for cardiomyopathy disease associations here, shown here in green. And I think a number of things are evidenced from this graph. So the first thing is that all of these genes have been associated in recent years, in the past 7 to 8 years, but they really are kind of recently associated genes. You can see that there's 18 genes now, with recessive inheritance, which is as many as for dominant inheritance with HCM or DCM separately. You can also see that the rate of increase is still quite strong, so it could be that we're expected to find yet more recessively associated genes in the coming years. But what you can see as well is in the past decade or so, probably most of the novel disease genes in cardiomyopathies are coming from these really rare recessive genotypes. Should mention as well that there was an additional 13 genes where we've got data currently just from a single case or from a single pedigree. So these are unreplicated. So these certainly will require further validation. Although important to say that for these 13 genes, most of the putative causal variants are missense variants, which is in contrast actually to the replicated genes where most of the causal variants are truncating. So it could indicate that within these 13 unreplicated genes, we might have a little lower confidence that these might actually be causal. So this just summarizes those 18 genes and the evidence we have underlying these associations. So it indicates a primary cardiomyopathy for each of these genes in the biallelic state, so DCM in blue, HCM in red, and arrhythmogenic cardiomyopathy phenotypes in gray. It lists the total number of reports where biallelic variants have been implicated and the total number of families within these reports. There's also some additional evidence, as I mentioned, where we have large pedigrees. It notes the maximum blood score. And also then for a couple of genes, NRAP and TRIM63, where we've got case control studies. There's also some additional supporting evidence, as I mentioned, from GWAS loci, where these are putative causal genes. So it's only for 2 of these genes where we see these potentially coming up in actual HCM or DCM GWAS. For most of the other genes, they're for related phenotypes, for like atrial fibrillation, or for relevant endophenotypes from ECG traits or left ventricular traits. Also noted as well, particularly for those genes where biallelic truncating variants are causal in the patients, we notice the mouse knockout phenotypes, and in general, these provide quite good supporting data. We see relevant cardiomyopathy phenotypes for some of these genes, or perhaps some of them lead to arrhythmogenicity or sudden death, and for a couple of them, they're all noted that they're embryonic lethal. So between all of this, we think we're quite confident in the pathogenicity of all of these genes. So looking at the clinical data, so it's going to summarize it for the 18 genes here. So on this graph is showing the age of onset for each of the patients along the x-axis. We also then note the particular variant class involved and the outcome in the patients. And then down the left, you can see the primary cardiomyopathy associated with these genes. So as you can see, and unexpected, I guess, for recessive inheritance, is that a lot of these genes are associated with really early onset disease, neonatal and pediatric cardiomyopathies. But there is quite a spread as well with some genes within genes, and also quite some genes that are at the top here, which tend to be more associated with more adult onset disease. So overall, as you can see, the primary phenotype tends to be predominantly associated with dilated cardiomyopathy. As mentioned, the age of onset tends to be young, but this is more so for DCM phenotypes as opposed to HCM. And the phenotypes are quite severe outcomes. So over half of these patients are reported to be deceased or required heart transplantations or insertion of LVAD. So I guess on discovering these robustly associated disease genes obviously also provides some good insights into disease pathophysiology and potentially novel pathways. So this is just a graphic which summarizes the potential cellular location and function of each of these 18 genes. So as you can imagine with cardiomyopathies, several of these tend to be associated with sarcomeric function, which is unsurprising. Several genes associated with Z-disc and with the thin filament. There's also some additional processes, perhaps more peripheral disease processes, where we do see these biallelic variants. Several of these are involved in proteostasis, for instance, like BAG5, which is a co-chaperone of HSC70, which is involved in protein folding, and also several genes associated with the ubiquitin proteasome pathways, which are involved in targeting sarcomeric genes, sarcomeric proteins, which perhaps have misfolded for degradation. We also see genes involved in the intercalated disc, for instance, those proteins encoded by MyZap and NRAP. So obviously, this biology is quite interesting, but how prevalent are these genes for cardiomyopathy patients? Are they ultra rare? Are there perhaps important causes that we should be testing for? So it's important to say for most of these genes, we really don't have good data for this. So most of the published cases are kind of single case reports or just reports from a few families. So we don't have really good cohort data for most of these genes where we could estimate the overall frequency. Now, in general, of course, we would expect the recessive cases to be much less prevalent than dominant cases. However, it could be that they are likely to be more prevalent for certain patient groups. So as I mentioned, they're likely to be more prevalent, of course, for pediatric cardiomyopathy cases. It may also be found more in certain diverse ancestry groups. It could be population bottlenecks, for instance, like Finland or Japan, or also regions of high consanguinity. And indeed, this is shown by this heat map here, where we show where we found some of these families, particularly those with homozygous variants. As you can see, these are particularly enriched around the countries of North Africa and the Middle East, where there might be more regions of high consanguinity, and you're more likely to detect homozygous variants. So one of the genes where we do have good data on the overall frequency is TRIM63. So this is a study from Spain a couple of years ago where they sequenced about 5,000 patients, majority of European ancestry, and found that biallelic variants in TRIM63 were found in about 0.3% of HCM patients. So this is in contrast to our study in the Egyptian population, where we found that these variants were found in about 2% of HCM patients in Egypt overall. So that's about a 6 to 7-fold increase within this kind of consanguineous population. So another way of estimating the potential prevalence of these genes and whether they might be more prevalent within certain ancestries is to look at the carrier rates within population databases like NOMAD. So the heat map here just shows you the percentage of individuals in NOMAD version 4, which you've updated since the paper, with heterozygous loss of function variants within these genes across the different ancestral groups. And here we're just looking at the genes where loss of function variants have been proven to be causal in the biallelic phenotypes. So as you can see from this, we see quite a divergence of carrier rates across different genes, but also across different ancestries. And there's a few, I think, interesting things to note about this heat map. The first of these is that NRAP, loss of function variants, seem to be particularly prevalent within the Finnish population. This is due to the fact that there are several loss of function variants present at low frequencies in Finland. And then this was confirmed in a recent study which found that biallelic variants within this gene are actually found in about 2% of all DCM cases from Finland. So might be particularly prevalent within this population. You can see as well that TRIM63 loss of function variant seems to be particularly prevalent within the Ashkenazi Jewish population. This is down to a single variant, which appears to be a founder variant within this population. And indeed, probably is so frequent in fact that I think it might be warranted for inclusion in carrier screening panels. We also see as well that there's a relatively high rate of these loss function variants within the Middle Eastern population. So it could be that the high frequency of biallelic variants we saw in our Egyptian HCM cases could be due to a combined effect of both this high background carrier rate and then also the effects of consanguinity. So of course, until now, we've been talking about recessive or dominant disease genes as quite distinct entities. But of course, in reality, as you might expect, it's much more complex than that, as you might expect from the title of the talk and the title of our paper. So if you look at textbook definitions of recessive and dominant inheritance, so for dominant, these would state that the biallelic phenotype would be exactly the same as the monoallelic phenotype. Whereas for recessive inheritance, it would state that you would get a phenotype with a biallelic genotype, but no phenotype at all with the heterozygous variant. Now, of course, in reality, as we would expect in clinical genetics and with disease genes, it's much more complex than this. And probably these variations on a semi-dominant model are much more likely to affect the clinical genetic reality. And indeed, this is being explored now in several different studies, including these papers here, which are biobank studies, which are looking at these spectrum of recessiveness and dominance within biobanks, in particular looking at the effect of heterozygous variants within recessive disease genes. And so within the cardiomyopathies, we also see this. I think one of the best examples is ALPK3. And so we've known for several years now that biallelic truncating variants within this gene are associated with a really severe or early onset cardiomyopathy phenotype. This is, in fact, was one of the earliest cardiomyopathy recessive genes to be reported. This is based on case reports from consanguineous families across North Africa, Middle East, and South Asia. Of course, then recently, Lewis and his team also reported the fact that heterozygous truncating variants within the ALPK3 are highly and significantly enriched in HCM cases compared to population controls, present in about 1 to 2% of all HCM cases. In these patients, it tends to be associated with the late onset disease, and these variants are probably present at fairly low penetrance within the population. So completing the spectrum, of course, then is the fact that common variants at the ALPK3 locus also shown to be strongly associated with both HCM and DCM and opposing effects. And so these are likely to be non-coding regulatory variants, which lead to minor changes in the dosage of this gene. So based on this, what we wanted to do with the 18 recessive genes that we found was to test whether any of these other genes had dominant associations with relevant phenotypes. So for this, we looked at the UK Biobank, looking at the 450,000 exome data, and looked for rare variant associations with the heterozygous variants here for 6 relevant cardiac disease phenotypes. We also then looked at the subset of the UK Biobank at the time that had cardiac MRI data. At the time was about 40,000 individuals, and looked for associations with various relevant quantitative traits, cardiac traits, which were related to function, volume, or hypertrophy. So this heat map just kind of summarizes those results. So you've got the 18 recessive genes along the y-axis here. Got the 6 cardiac disease phenotypes, and then also the 11 MRI traits. So as expected, we were able to validate the association of heterozygous loss function variants in the LPK3, HCM, validating what Lewis saw in his paper. Also noticed some novel associations, association of a heterozygous loss of function variants in LMOD2 with DCM, which is consistent with what we see with the biallelic phenotype, but also in association with heterozygous loss of function variants in LPL3L with atrial fibrillation. There are also some putative associations. Obviously, the significance here is much less. It's kind of more of a nominal association, and these would need to be repeated, of course, and validated in other data sets. But it still kind of makes sense, and it's consistent with the biallelic phenotypes. So in this case, it was associations with heterozygous loss of function variants in SLC30F5 and KLHL24 with HCM. Also potentially some interesting signals within the quantitative MRI data. Obviously, we're quite underpowered to really detect very significant associations here, as there was only 40,000 individuals at the time. Nonetheless, some of the potential signals I think are quite interesting, and again, tie in with the biallelic data. So for TRIM63, so as I mentioned, biallelic loss of function variants here associated with hypertrophic cardiomyopathy. Indeed, here we see for monoallelic loss of function variants, they seem to have some sort of signal associating with increased hypertrophy, both for LV mass, and also septal thickness. And likewise for JPH2, where biallelic truncating variants are associated with DCM, where these heterozygous loss of function variants seem to be associated with a reduced left ventricular ejection fraction, again, consistent with what we see with the biallelic variants. So this might indicate that for certain genes, while we might not see a dominant association with the disease phenotype as such, they might be having some subclinical effects which could contribute to the disease etiology. So just going back to the biobank data sets that I showed you earlier, where we found the known cardiomyopathy disease genes. So LMOD2, which we discovered this UK biobank analysis. If you look back at this river association study for primary intrinsic cardiomyopathies, this in fact is located just underneath the threshold for genome-wide significance. So I think this kind of indicates how kind of a knowledge of these rare recessive genotypes can be used to inform the discovery of these dominant associations, and can be used to distinguish the potential signal underneath genome-wide significance from the potential false positive hits. There's obviously other orthogonal data sets you can use for this as well. We can use things like GWAS data or animal model data, which might also be useful, but I think the recessive human genotypes are probably potentially the most relevant for this task. So this is a model we kind of proposed as well for us potentially for looking at the relationship between the biallelic and monoallelic genotypes for these genes. And we think there might be some sort of association between how severe the phenotype is for the recessive inheritance and the likelihood of seeing a phenotype in the monoallelic state. So we think if we see a really severe phenotype with the recessive inheritance, so really severe early onset pediatric disease, we're probably more likely to see an association with heterozygous variants, as is the case for ALPK3 and LMOD2. Whereas for biallelic variants that cause more adult onset disease, you're probably less likely to see any signal with heterozygous variants, or if you do, it's probably maybe just going to be a subclinical phenotype and a risk factor. So in a practical sense, should we be including these genes in clinical diagnostic gene panels? Overall, I would say yes, I think it's quite important. And important to say that several of these genes could already actually be on these cardiomyopathy panels, so genes like JPH2 or LDP3 due to earlier proposed monogenic associations. But of course, it'll be important to be aware of the distinct phenotypes for monoallelic and biallelic genotypes, and also for missense or loss of function variants. So of course, individually, these 18 recessive cardiomyopathy genes are probably likely to be rarely causative in most patient groups, but collectively they could perhaps explain a not insignificant portion of cases. And as I mentioned, they might be particularly important for certain diverse patient groups and ancestries. So I think if you're currently looking at virtual panels, that is, you're doing exome and genome sequencing and just doing a test based on a subset of that data, so it'll obviously be quite easy just to add these genes to that test. For actual physical sequencing panels, it's obviously more complex, particularly due to the increasing list of gene associations and the constant need to redesign these. I think one advantage of including these, though, is that we will generate over the next few years then obviously large clinical genetic data sets. This would allow for accurate estimation of the prevalence of these recessive gene types, but also further assessment of any potential mononucleotic associations and potentially to try and validate some of these signals that we're seeing in the UK Biobank. I think important as well, though, that as we begin to increase the size of these or clinical testing panels, again, obviously it'll be important just to report on the relevant genotypes. So for example, if a gene is associated with cardiomyopathies just with biallelic truncating variants, it probably doesn't make sense just to report every heterozygous missense variant as a VUS, as that would just create kind of noise and uncertainty around the clinical genetic testing. So the final aspect I just wanted to address was looking back at the cardiomyopathy genes of primarily dominant inheritance. Obviously, these are the most prevalent cardiomyopathy genes. This is where we find most of the cause of variance in our patients. And so we just wanted to check as well, do we also see a spectrum of zygosity effects within these genes? Now, of course, over the years, there have been occasional reports of biallelic or homozygous pathogenic variants within these established genes. So perhaps one of the most common being truncating variants in MYBPC3, which in the biallelic state tends to be associated with neonatal and severe pediatric cardiomyopathy. So these kind of reports would suggest that these kind of semi-dominant models are also relevant for the primarily dominant cardiomyopathy genes, but the effects, of course, are likely to vary by gene and variant class. So with our meta-analysis, we also wanted to look at these genes. So we also noted the biallelic genotypes that were found in the genes with primarily autosomal dominant inheritance. So in total, we found about 200 reports across 27 genes. And again, we might have the information here around the clinical phenotype and outcomes, the demographics, but also here paying particular attention to the status of the heterozygous relatives and whether they were affected as or not, because you might certainly expect some of them to be affected given the fact that these are genes primarily associated with dominant inheritance. Now, some of these genes obviously have quite distinct syndromic or non-cardiac phenotype associations that are well known. So, for instance, biallelic missense variants in genes like titin and lamin are known to be associated with kind of skeletal myopathies. We know, of course, that variants in desmosomal genes, which are associated with ARBC, in the biallelic state tend to cause the syndromes Naxos and Carvajal syndrome, these cardiocutaneous syndromes. So for this, we decided to focus more on the sarcomeric gene variants associated primarily with HCM and also with DCM. This is where we had the largest data set of biallelic variants. So before I go into the results of that, I'll just mention some of the key results that we found from our study in the Egyptian population. So as a reminder, in this study, we looked at over 500 HCM cases. Important aspect of this is that 28% of these patients were reported to have consanguineous parents, that is first cousin parents. So obviously you would expect these to be enriched for homozygous variants. So in this study, we looked at panel sequencing data across 13 genes which are associated with HCM with autosomal dominant inheritance. In total, 6% of all the cases had homozygosity of rare or low frequency variants in these autosomal dominant genes, as compared to 0% in population match controls. The interesting aspect of these results is that these homozygous variants were not evenly distributed across all of the HCM dominant genes, but rather were particularly enriched in a subset of these. So these are missense variants in the myosin light chain genes MIL2 and MIL3, and truncating variants in the CSRP3 gene, which codes for muscle limb protein. So this, of course, might indicate that some of these genes of varying classes could be more likely to express the phenotype in the homozygous state, indicating as well that they might be less penetrant in the heterozygous state, and again, indicating more of a semi-dominant model of inheritance. So going back to our meta-analysis, in total, we've identified 79 cases across 75 studies, and notice as well that there were some quite distinct associations by gene and by variant class. So we did across 8 genes, 7 sarcomeric genes, and also CSRP3. So focusing initially on the biallelic truncating variants, so these are the full knockouts. So 3 of the core genes, MH7, TNNT2, and TPM1, didn't find any clinical reports. This might indicate that these are probably incompatible with life and embryonic lethal. And indeed, this would be supported by the fact that all of these gene knockouts in mice are non-viable and tend to die as embryos. So for some of the other key sarcomeric genes, MYBPC3, TNNI3, MIL2, and MIL3, biallelic truncating variants in these do tend to lead to severe early onset disease with some mixed cardiac phenotypes. In contrast, and as we saw with the Egyptian data, biallelic truncating variants in CSRP3 unusually lead to more typical adult onset HCM, despite the fact that it's a full gene knockout. So moving on to the non-truncating variants, largely missense variants, as you would expect, and these tend to lead to less severe outcomes. So on average, they present in young adulthood, so probably somewhere intermediate between the biallelic truncating variants and your normal heterozygous missense variants. So in terms of age of onset, there's not a lot of difference between these genes. Where we did notice a difference was in the proportion of relatives who are heterozygous who are reported to be affected. So for some of these genes, particularly TNNI3, MIL2 and MIL3, none of the heterozygous relatives in these studies were reported to be affected. So again, this could indicate that these types of variants have a lower penetrance and effect size in the heterozygous state. So this ties in well, I think, with what we saw with the Egyptian data for MIL2 and MIL3. And it also ties in well with a study from the Bart's group, recent study which looked at the penetrance of these missense variants in sarcomeric proteins in family studies, which found that carriers of TNNI3 variants were significantly less penetrant than the other sarcomeric genes. So I think overall, this kind of shows you that the analysis of these really rare recessive genotypes, rare biallelic phenotypes, can be used to address one of the key issues around cardiac genetics, which is trying to understand the degree of penetrance in relatives and in the population. One final study I'd like to mention, which is recently published by Juan Pablo Ochoa and his group in Spain, which is slightly more unusual in that it doesn't deal with a rare variant, but rather with a low frequency variant. And this is a study that looks at missense variant in FOD3, RH637 glutamine, which is found in about 0.6% of their controls and in Europeans in general. They found that this variant is highly enriched in HCM cases, significantly enriched, with an odds ratio of about 5, which indicates that in the heterozygous state, it probably acts as an intermediate effect variant within HCM patients, contributing to disease, but obviously not primarily causal. They did find, however, that there were 5 families where the variant was present in the homozygous state. And in these families, the homozygous patients, had a really early presentation and a relatively severe phenotype. So again, kind of showing the spectrum of effect. So I think FOD3 is probably one of the most interesting cardiomyopathy genes looking at this allelic spectrum. So we map the various different variant classes in FOD3 along this kind of standard graph of comparing effect size and allele frequency. We would see that these really rare variants, the rare missense and exon deletions, particularly those around this pathogenic hotspot here, have very high effect sizes and tend to act as pathogenic Mendelian-like variants. At the other end of the spectrum, we know that common variants at the FOD3 locus are robustly associated with both HCM and DCM with opposing effects. And then in between these, you have this variants like this RH637 variant, which is present at low frequency in the population and acts with an intermediate effect size. But of course, we can also superimpose now the spectrum of zygosity as well in here. And we can see that in the homozygous state, this intermediate effect variant actually acts much more like one of these rare pathogenic Mendelian-like variants. So just to sum up, I think we've seen that recessive disease genes are obviously becoming a much more important aspect of cardiomyopathy genetic architecture. It seemed to account for most of the novel gene discovery in cardiomyopathies in recent years, and certainly have helped to expand our understanding of disease pathophysiology and pathways. As you've seen as well, kind of simple textbook models of dominant or recessive inheritance often do not reflect clinical reality. So many of these disease genes do demonstrate some sort of allelic spectrum of effect. I think for clinical application and for understanding the clinical effect of heterozygous and homozygous variants, it'll be important to understand all of these at a gene-specific level. I think, as we've seen from this as well, that research in previously understudied groups is really essential both for gene discovery and understanding genetic architecture of cardiomyopathies, but also for increasing equity for clinical genetic testing in diverse groups. And also how discovering and characterizing what might be really rare recessive disease genes can also aid in the discovery of potentially the more common dominant forms. I think it's particularly important for relatively rare diseases like cardiomyopathies, which probably are currently underpowered for discovering the rare genes left to discover through things like rare variant association studies. Just like to mention as well, so recently we've been producing some infographic gene summaries around these cardiomyopathy recessive genes, which just highlights some of the evidence underlying these associations, a quick clinical overview in terms of age of onset and outcomes, any particular ancestral associations, and also whether we see any monoallelic associations for these genes. we've been producing a couple of these recently, BAT5 and LMOT2. I'm producing a few more of these in the coming weeks. So if you're interested in seeing these, just have a look on Twitter and under the hashtag recessive CM genes. So again, just like to acknowledge the people involved in these studies in Amsterdam, Connie, Alex, and Sean, and our colleagues at the Broad Institute as well. Of course, at the Magda Yacoub group, Professor Yacoub, Yasmina Mona, colleagues at Imperial, and also Francesco in Italy. Of course, doing studies like this, meta-analysis study like this, is obviously completely dependent on studies which have been already published. And so this just lists all of these studies looking at the kind of rare recessive forms of cardiomyopathies. I think for looking at all of these kind of rare recessive genotypes, diseases like this, obviously these kind of studies are essential, these kind of single case studies and single family studies. And so I'd just like to acknowledge these here. So thanks for listening. I'm happy to take any questions.
Thank you.
I think that was a masterclass in the spectrum of genetic cardiomyopathy. Thank you, Roddy. Just to say to everyone, they can post their questions using the Q&A function of the webinar, or they can raise their hand and Luis.
I wasn't sure if you had finished because there was some sort of pause there.
Sorry.
Now I was going to say thank you as well to Roddy. This focus of the talk, which was amazing, very clear, very thorough for a relatively complex matter. But I really enjoyed the focus on this recessive field, which we often obviously hear about autosomal dominant models. Lately, there's a huge interest in common variant effects and the interaction between common and rare, but this whole field of recessive, as you showed very well, has suggested also new genetic etiologies and a new way to look into gene discovery, but also important translation points that are already being applied in terms of what genes to report and what genes to test in terms of clinical genetic testing. I'll just maybe start the discussion with a couple of comments and questions. The first one, I mean, it's quite humbling to see the ARVAs that you showed, because usually when we started to do whole exome sequencing and have access, particularly to a couple of thousand samples, I think it was the general feeling of the community that we would be able to, just start finding more and more candidates, at least candidate genes for cardiomyopathies. And again, in a humbling way, we figured out that that was a bit more challenging and tricky than it seemed at the beginning. Obviously, some signals we did get from doing that kind of burden testing work. LPK3 was one of the occasions that we've managed to do that, but as you very well showed, it's quite tough. And that very nice work that you showed with the RVAS graph similar to the GWAS, but for rare variation, very neatly shows why it is so challenging. I have a very brief sort of technical question. The RVAS, is it done like a burden testing as an aggregate of rare variants for each gene, or is it done at the variant level like a GWAS, let's say?
No, yeah, it's an aggregate level. So it takes all the variants, the rare variants within a particular gene, and looks at in cases versus controls. So they tend to look at different kind of variant masks, they call them. So sometimes you can look at both loss of function variants or missense variants, or missense and loss of function together. And so you can look at various different masks to see where you might find the best signal. So I think a lot of the time, loss of function masks, looking at loss of function truncating variants only, can be a little bit more powerful because they tend to be lower in the background in the general population. So you tend to have a bit better signal, like with the LPK3. But yeah, it's those kind of cumulative tests.
Because it's, I mean, with all those thousands of cases and controls, and still it's always the same suspects that become highlighted in terms of odds ratio. But this type of other approaches that you've shown hopefully will generate more knowledge. And
yeah, I mean, I think there's a lot of data in there, but like, if we're trying to find rare causes of relatively rare diseases, then we're obviously still underpowered. So we'll need to perhaps not just do a simple draw the line and only look above it, but perhaps you do slightly more sophisticated analysis by looking at the kind of subgenome-wide threshold as well and trying to integrate different lines of evidence.
Absolutely. And the other second highlight for me is this, which I was thinking about before you showed your one of your last slides, about the intermediate variants, which we always wondered about. We have a huge number, obviously, of those on our databases. The work from Juan Pablo is very interesting. He picks up a specific case of this relatively common, although in intermediate spectrum variant, with a clear effect as an homozygosity. But I was wondering, in terms of strategy, as a community, as a research community, to explore intermediate variants, what do you think is the best strategy to do it?
To identify these, so obviously you need big data sets and to kind of demonstrate we would expect to see an enrichment in cases versus control. So obviously you need a big data set to try and kind of demonstrate that and to try and identify those. Obviously you want to validate them as well then, ideally with maybe functional data, although it can be tricky because you might see a very clear functional effect. So previous studies we've done, we found 2 of these types of variants in troponin genes, which are present in Chinese ancestry cases. And we were able to kind of demonstrate an effect on MRI phenotypes for one of the variants. And we also did some sort of experimental validation of the other ones. But obviously that's a lot of work to try and validate these types of genes, these types of variants. But yeah, it's a lot of work to do that. But I definitely think we need to start paying more attention to these. Particularly as I think when we're trying to develop kind of more comprehensive genetic profiling of patients, I think in the next few years we'll try and integrate, the rare Mendelian-like variants with your polygenic risk scores to try and develop an overall genetic risk profile. And so I think we're going to move towards that. We need to not forget about the low frequency variants as well. Otherwise, the PRS just won't work as well because we'll have a lot of noise in there.
But then you are looking just at the 2 extremes and a lot of other stuff in between.
Exactly.
But there's a question from Juliana Lores. She's asking if you have any data about troponin C homozygous variants in early onset cardiomyopathy.
I think there was maybe only 1 or 2 patients came up in the meta-analysis for TNNC1 from memory. So probably wasn't enough to actually include in the meta-analysis because we couldn't really draw too much from that. I'd have to have a look again, but there wasn't much. Obviously, it's a very rare gene just for heterozygous variants. We see very few patients in general with TNNC1. So they tend not to come up, unfortunately. We certainly didn't detect any within our Egyptian cohort either.
Thank you. Does anyone want to? We have quite a big audience today. Alex, Max, Alex wants.
Yes, I guess I can. I find quite interesting. I was involved in the CleanGen project for the GCEP in ARVC and DCM, and I think it was quite difficult with some papers because we had to go by the phenotype reported. We didn't actually have the actual phenotype of the patients to decide what kind of disease they have, and I just wonder whether there are any kind of novel ways or a tweaking of the current ways that we can do this analysis to incorporate the spectrum of the phenotype as well, rather than just having disease yes no kind of a feature. What do you think?
You mean within the kind of ClinGen evaluation?
No, as in general on assessing the role of these variants, because now we're looking at specific diseases, whether they're present or not. But in reality, the phenotypes are even more gray than the genetic background. So how do we navigate that in the future?
I mean, that's certainly, I think, the advantage as well of these kind of rare variant association studies. So you can kind of look, rather than looking at a specific disease or a specific ICD code, you can look at the gene level and see what kind of associations you're seeing across a bunch of various different phenotypes. And sometimes you see that it's sometimes it's quite specific to a certain disease, but oftentimes you see a lot of potential associations across different, for instance, cardiac phenotypes, but also non-cardiac phenotypes, which kind of might indicate that these are a bit more complex. And also with the recessive cases, I mean, it is, as I mentioned, quite complex to kind of totally tie down as to whether many of these are very specific to cardiomyopathies as such. There's many genes which are identified, which are associated with more syndromic conditions, multi-system disease. Some genes which are associated with what I think are syndromic conditions, but can sometimes present with cardiomyopathies, kind of genocopic conditions, again, Pompe and Friedreich's ataxia. And so drawing the kind of very clean dividing line between those is not easy, but it's something we'll have to look at more in the future.
Yeah, and I think there's probably some underreporting also on, heterozygotes from recessive diseases on their, let's say, disease burden. I mean, we've had this issue in Axis disease, for example. The heterozygotes, they've not been, studied to the extent of the homozygotes, and that definitely makes it more unclear when you do meta-analysis of these studies.
Yeah, absolutely.
We have two more interesting questions. One from Sabine Klassen, highlighting the importance of ethnicity in the interpretation, particularly of homozygous variants, and asking if databases like ClinVar can account for this. And Antoine Bondieu is asking about the skeletal muscle involvement for the recessive variants, if it is more frequent than usual.
So in terms of ethnicity, yeah, I mean, obviously it's essential. So I think it's essential just to have much more diverse data sets to begin with. And obviously databases like Nomad are essential for that, but also obviously there's entries in ClinVar which can describe these as well. I think Nomad's getting better. So as I showed you there, we've got a kind of a Middle Eastern, reasonably good sized Middle Eastern data set as well. But obviously our studies in populations like Egypt as well is very important for this and for kind of interpreting these. And so I think we just really need to expand these more and more, and not just expand like cardiomyopathy genetics research into other populations, but also continue to create these population databases.
Absolutely. And the other question was about the skeletal muscle involvement for recessive variants, if it is more frequent than usual.
Again, it kind of depends what recessive genes you look at. So I think, as far as I remember, for the genes we kind of selected for the analysis here, and the 18 kind of cardiomyopathy associated genes, I didn't notice anything particularly specific around skeletal variants. But of course, there are a lot of other ones that are kind of slightly more syndromic in nature, where you might have both cardiac involvement and skeletal muscle involvement. But we didn't they weren't the primary focus of the analysis. So, but there's plenty of other genes in there, and within the supplemental tables where I think there probably are ones with joint cardiac and myopathies.
And then there's an anonymous question about mitochondrial variants, which is, well, another very complex world in terms of absence, even of, sometimes controls and proper frequency in the population. Do you want to take that one?
Yeah, I can do. I mean, it's not something I've kind of looked at much or know too much about. I would say we've recently completed the ClinGen reevaluation for HCM. The second round of that was led by Jody Ingalls. Yeah. There was one particular mitochondrial gene which we looked at and decided had, I think, at least moderate evidence of association. So that's one of the mitochondrial genes, which I think should be included certainly for HCM genetic testing now. But beyond that, I don't know too much about it.
Yeah, I think there is a tendency to include some of the mitochondrial variants, like the 3243 or the 4300.
Yeah, exactly.
HCM phenotypes. There's another question also highlighting the importance of ethnicity, which we might close with this one. And actually, when you were commenting, and this is also commented here in this question. So the question is, because I don't know if everyone can read it, if we think improving data gathering from underrepresented populations could affect the results, which we already kind of touched upon. And the she gives the example of Latin America, and also mentions recent work by Jody Inglis, which I was just thinking about on, I think it was about sort of New Zealand or Pacific population.
Yeah.
Probably saw that. I think
Yeah.
she basically published, and a variant that was thought that was rare was actually quite common, and the classification changed completely. But yeah, it's definitely very important to do more and more work, and gather more and more sort of controlled general population data in this in all ancestries and all ethnicities.
Yeah, absolutely. I mean, that's about as the question says, kind of taking a high number of the US and trying to resolve them in the terms of downgrading them. Absolutely. And so that's what additional genomic data sets and diverse genomic data sets can certainly do. I think it's probably particularly important more for the kind of, I guess, the dominant disease heterozygous variants. Certainly for the recessive ones, I think it's a little more clear, particularly for the bilated truncating. We can be quite confident that those ones are at least causal. But yeah, it's definitely an issue, and we need more and more diverse data. Absolutely.
Great. I don't know if we have time, Alex, for other questions, or do you want to close?
I think there's just one more from Talia Patterson. You can read it. Can you see it?
Yeah, it's related to one of the last bits of our discussion. If the importance of intermediate effect variants versus polygenic inheritance in HCM not clear yet, if you want to elaborate about it.
Yeah, so I mean, these variants they're, as the name suggests, intermediate between your kind of our rare pathogenic Mendelian-like variants and the kind of common SNPs that we identified through GWAS. So, if you've got odds ratios somewhere between 5 and 10, it's obviously going to contribute to the disease in some sort of way, but it's obviously not primarily causal, and the penetrance in the general population is going to be quite low for all of these. But certainly within families and within patients, it's probably going to act as a bit of a modifying factor, either in addition to, say, a high PRS or in addition to a primary pathogenic variant. And so I think it'll be important to identify these, to characterize these, and then to try and eventually kind of include them into clinical genetic testing in terms of kind of looking at the overall risk profile in patients and their family members. Might kind of inform, whether for currently healthy individuals and within families, whether they have the additional hits might indicate they're more likely to get the disease and should be monitored more closely. But yeah, overall, as I said, as we kind of begin to develop more comprehensive genetic risk profiling, we'll need to definitely take these into account as well.
Very, very, very quick last question. Are you still, with all this variability, with all this complexity, and obviously I know where the numbers come from, but are you still comfortable with the 1 × 10⁻⁴ filtering threshold?
Yeah, that's a good point. It depends what you're looking for, I guess. If you're looking for primary pathogenic Mendelian variants, then yeah, you should be looking at that level. Obviously, you don't want to just say, well, we've seen things like ARG637, the FOD3 variant, therefore we should just relax the threshold completely, because most of the non-rare variants are not going to be doing anything. So I think it's important to stick to kind of stringent thresholds where you're most of the variants you find, but then specifically identify the ones which are less rare, but potentially are associated with disease. But yeah, it's challenging.
Yeah. Thank you so much.
Great, thank you.
Want to close?
Yeah, I think we can close the session. It was really great. It was very interesting talk and discussion. Thank you, Roddy. Thank you, Luis, and thanks everyone for joining. And we hope to see you soon again.
Great,
Take care. Have a good weekend. Bye-bye.
Bye.